Alright, so in this video we're going to use the not so popular board game Mousetrap to model cellular respiration. It seems a little weird at first, but if you follow along, I'm sure you'll enjoy this. So let's just review. Glycolysis occurs in the cytoplasm. The goal of this is to take glucose and form NADH, electron carriers. That will eventually go to the electron transport chain to create ATP. The pyruvate from glycolysis enters the mitochondria to go through the Krebs cycle, which makes way more electron carriers, all of which go to the electron transport chain to make ATP. Note that we do a little bit of ATP, we make a little bit of ATP from glycolysis in the Krebs cycle via substrate level phosphorylation, while in the electron transport chain we make a lot of ATP with oxidative phosphorylation. So let's begin with the model. So what we have here, sort of labeled but is kind of hard to see, is we have NAD+, which are represented by little balls of aluminum foil that are supposed to be marbles. So what we do is we load the NAD+ into the bucket. This bucket is NAD+. And so we're going to use the boot, which is glucose, and we're going to oxidize the glucose. So me pressing down on this is the oxidation of glucose. So we begin oxidizing glucose, which kicks over the bucket, and now we have NAD+, which is going to get reduced through glycolysis in the Krebs cycle to NADH. So there's glycolysis, there's the Krebs cycle, boom. There it is. We have NAD+ at the end of the alligator, which is the Krebs cycle. So we're going to do this multiple times. Glucose makes a lot of electron carriers, reduces a lot of electron carriers, right? So as we can see from our little chart, we're actually going to get 2, 4, 10 NADH and 2 FADH2 from these processes. So we're not going to do that many electron carriers for the sake of the model because we don't want it to take too much time. But let's say we'll do 3, because 3 NADH is enough to make an ATP in the electron transport chain. Alright, so here we have 3 NADH. It was NAD+, but we reduced it through the power of glucose oxidation through glycolysis in the Krebs cycle. So now we're at the giraffe. So the giraffe is oxygen. Oxygen is the final electron acceptor. So oxygen doesn't necessarily carry the NADH to the electron transport chain as we're going to say it does here. But what it does do is it pulls the electrons from NADH down the electron transport chain because it's the final electron acceptor. So oxygen is sort of the barrier between the Krebs cycle, or Krebs and glycolysis, to the electron transport chain. Because if oxygen is not present, then there's nothing to pull the electrons down the electron transport chain. But in this case we do have oxygen. So these get to go through the electron transport chain and down through to create the proton motive force, which is our hippo that sits upon here. So after oxygen pulls down the electrons from the electron transport chain, it's going to go down, 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 and it collects. It creates this proton motive force, and then this should be reset. It's kind of finicky, but let's say that it's right here. So the proton motive force is going to power, on the bananas, ATP synthase to trap ATP, which is the cheese. So this whole model is set up to show that the reduction of electron carriers is done to collect energy from the proton motive force by ATP synthase to make ATP in the cell. Note that oxygen needs to be present so that the electrons can be pulled down the electron transport chain. So if there's no glucose, there's no energy, right? There's no energy to push the bucket over to reduce the electron carriers, right? If we run out of electron carriers and there's nothing left in the bucket, then we can no longer we can no longer oxidize glucose and reduce electron carriers. So if there's no oxygen, what we're going to end up, so say the oxygen isn't present, the giraffe is gone, we're going to reduce all these electron carriers, and they're just going to build up down here because no oxygen is available to take it to the electron transport chain, quote unquote. Right? So we'll just have these build up. So if oxygen isn't present, our giraffe is gone, then what is a cell to do? That's whenever the fermentation mouse comes into play. What fermentation essentially does is it takes the electron carriers back to the bucket, right? So that's how we show fermentation. It's going to regenerate NAD+, right? Because if we have too much NADH built up and no oxygen to pull the electrons down the electron transport chain, then the cell won't be able to do glycolysis anymore because it needs NAD+ to oxidize the glucose. So if we're going to keep on doing glycolysis, then fermentation is needed to regenerate the NADH. So that's what happens here with fermentation mouse. Alright, so say that oxygen is present and we are forming all these electron carriers, but we introduce, let's say, carbon monoxide, which is a respiratory inhibitor. So this is going to block the electron transport chain. In reality, it is a protein inhibitor. It's going to stop one of the proteins of the electron transport chain, but here we see that it's blocked up. So oxygen pulls it up, and nothing can get through. So this is carbon monoxide, a respiratory poison. So that's the wrong marble that enters the electron transport chain. Another respiratory poison is cyanide, right? So hopefully from this, you sort of get the big idea of cellular respiration. It's easy to get bogged down in all the numbers and all the terms, but this is what you should really know, is that glycolysis in Krebs is to reduce electron carriers from the oxidation of glucose. They're coupled together. Oxidation must be coupled with reduction. And that oxygen's primary purpose is to pull these electrons down the electron transport chain. And if oxygen isn't there, then fermentation is needed to regenerate those electron carriers. Now, all of this is done to trap ATP using the proton motive force and ATP synthase.